Hey everyone, my name is Yinwei Xia, and I'm part of the DKU class of 2028. This is my Biology 101 presentation. Today I'm super excited to dive into a fascinating topic in biology, the synthesis and secretion of proteins. Have you ever wondered how cells make proteins and send them out to do their jobs? It's a process that's both complex and amazing, and I can't wait to let you go through it. So let's start from the brief overview of the secretory process. We have three steps here. There are the ribosomes, polypeptides, and then, lastly, the proteins. So, all right, let's start at the beginning. The story of a protein synthesis begins with ribosomes. Think of ribosomes as the tiny factories inside your cells. They are made up of two main components, ribosomal RNA, and you can call it rRNA, and ribosomal proteins. The rRNA is produced in a special part of the nucleus called the nucleolus, while the ribosomal protein are made on the ribosomes themselves in the cytoplasm. Once they come together, ribosomes are ready to do their jobs. So what do ribosomes do? They read the genetic code and assemble amino acid into long chains called polypeptide. It's like building a necklace with beads, except in this case, the beads are amino acids and the necklace are polypeptides. The polypeptide is just the start, the generating just start. And the polypeptides need more processing before they become functional proteins. Lastly, we come to the rough ER part. Oh no, it's not the lastly, but the next step. So once the polypeptides are made, they head over to the ER. The ER is like a mat of tunnel inside the cell, and there are two types, the rough ER and the smooth ER. The rough ER is called rough because it's covered with ribosomes. This makes it look like it has a tiny box, and this is where the polypeptide go next. So in the rough ER, the polypeptide undergo some important changes. They get folded into right shape and sometimes even get modified with other molecules. And this is crucial because the shape of protein determines its function. So think of it like origami. If you fold paper in the wrong way, it won't work as intended. So the RER ensures that our proteins are folded just right. Then we go to the Golgi. After the rough ER, the proteins are sent to the Golgi. The Golgi is like the post office of the cell. It takes the proteins, further modifies it if needed, and then packages them into little bubbles called vesicles. And these vesicles are like very tiny delivery trunks. They are ready to transport the proteins into their final destination. Then, finally, the vesicle carries the proteins reach the cell membrane. Here, they fuse with the membrane and release their contents outside the cell. The process is like this. And this is a vesicle, this is a membrane. To get to the membrane, then there's some proteins of some things that are released. So it's like the proteins are being shaped out of their cells to do their jobs. So for example, insulin, a hormone that helps regulate blood sugar, it's a secretory protein that gets sent out of the cell to do its work. Now, let's talk about the magic behind exocytosis. The cell membrane is an amazing structure that they have two properties. The first one is fluidity, and the second one is polarity. The fluidity of the membrane allows the lipids and the proteins to move around, making it flexible enough for vesicles to dock and fuse smoothly. Meanwhile, the polarity of the membrane ensures that the vesicle fuse at the correct location, maintaining the cell structure and function. So these two properties work together to ensure that the proteins are released efficiently and accurately. And we come to the conclusion part. So that's the journey of secretory protein from the start points to the finish. So from being made in the ribosomes and being shipped out of the cell, every step is really essential. Cells are like tiny, well-organized factories, and the protein are the workers that keep everything going smoothly. So I hope you enjoyed today's video and enjoy this little tour of the cell's protein synthesis process. So thank you for your watching.
So, firstly, I want to express my thankfulness to my professor, biology professor, and Professor Lee. Your encouragement has been valuable, especially during those moments I felt less confident in my ability. You have a remarkable understanding of the challenges faced by the freshman students just like me, who especially come from a weaker background in biology and feel apprehensive about venturing into unfamiliar territory. Also, your prompt and thoughtful responses to my emails, even late at night, have truly touched me. Thank you for being such an inspiring and supportive mentor. And because you're Korean, so maybe I'm willing to use some Korean words to express my thankfulness. And okay, it is 감사합니다. I'm not sure if I pronounce it right. So yeah, this is for my professor. And then I may expand my thanks to all the teachers and classmates who have contributed to the Biology 110 course. I will always remember the patience and dedication of the lab instructors who generously shared their experience in the lab with me. Their willingness to help, even when I struggled, was truly inspiring. Also to my classmates, you make me be supported, and that means so much to me. So your kindness and encouragement have made this journey so much more enjoyable and rewarding. So this is a challenging course, I have to admit, especially for me, but I still get some valuable experiences and thinkings. Thank you for all your efforts, patience, supports, all the things.